Okay, coiling tube, and it is located like this. If I am standing in front of you, my two cochleae are like this. Okay, one is here and one is here like this. Okay, two rings are there projecting you anteriorly like this. As this is the shape. Okay, now cochlea is a coiled tube, bony tube, and inside that tube you have the membranous pipelines or the membranous tubes module. This is coiled. If you just open up and make it straight, it will become like a straight tube. Okay, for understanding you can make it straight like this, then you can recoil it. Now if you make it straight, it becomes like a single pipe, and in that single pipe three pipes are maujood. Okay, one pipe is called the scala vestibuli, the other pipe is called the scala tympani, and the third pipe in between is called the scala media. So three pipes are maujood in one bony tube. Okay, now if you fold this bony tube, the pipes inside are also folded like this. Okay, so there is one fold, complete fold, there is second fold, complete fold, and there is third fold, partial fold, or it is 3/4 of a turn like this. So there are not 3 turns, less than 3 turns for the cochlea. When you are having one fold, one turn, this is the biggest turn. The second turn is a smaller turn, and the third turn is the smallest turn. So it means you are going from big area to the small area. So the tube, the shape of the cochlea is like this. It will narrow. This part is the apex of the cochlea, okay, and this part, starting part, is the base of the cochlea. Okay. Now, where is cochlea maujood? In the middle ear cavity? Okay. Now, let us talk about, let's take our concept here very quickly. What window is that? What window is here? And the rest is bone. Sir? The tube of cochlea is present. This is the bony wall, and from here the cochlea is starting. Okay, just behind that, it's the straight tube of the cochlea running like that. I am making it straight in total. Okay, it's running like that. How many pipes are maujood in the cochlea? 3. Okay. Now, if you remove the stapes bone, okay, remove it, and you see what is there inside, you will go into the scala vestibuli. Okay, which tube you will go in? Scala vestibuli. That is filled with a fluid which is known as perilymph, like CSF. Okay, perilymph. Alas. Okay, so this tube is scala vestibuli when you remove the oval. Now, you remove this membrane and you look into this one. There is another tube going. What is this tube? Scala tympani. This tube is scala tympani. The scala vestibuli and scala tympani, when they are going at two ends, they meet each other like this. And there is a connection between the two tubes. That connection is called helicotrema. That opening is called helicotrema. I'll show you that. Look at that one. This is a straight tube. This is oval window. This is round window. In front of that you have scala vestibulus. Here you have the scala tympani, and these two are meeting at this point from inside, and that point is known as the okay. This is the base of the cochlea. This is the apex of the cochlea. So if you are making it one turn, second turn, like this. So this part will come on the top here, like this. Okay, this is the apex. Helicotrema is the apex of the cochlea. Okay. There is another tube which is maujood in between the two tubes. Okay, but where is that tube? That is the scala vestibuli there. That is the scala tympani there. And here you have the bone. So what you do? You take a hammer and you break this bone. Remove this bone. Now you will see a tube, because it is bone here. Behind this bone, a tube is running maujood, which is also filled with the fluid. That tube in the middle is known as the scala media. So now you have got 3 tube systems which are maujood there. Okay, vestibuli, media, and the vestibuli and tympani are connected to each other. Yes or no?
Yes.
If they are connected, should they have the same fluid or different fluid?
Same.
What is that fluid? Look at the scala media. Closed on this side, closed on the other side. It's a sealed tube on 2 ends. Should it have a different fluid or the same fluid? Different fluid. And what would that be? Endolymph. So perilymph, and in between you have the endolymph. But endolymph is not mixing with the perilymph. Perilymph is continuous in the 2 cavities. but endolymph is totally separate. Are you getting the design of the system? All right, is it clear so far? Now, very quickly, let us see how are these tubes separated. There is a membrane module here, upper membrane, and there is a membrane module lower. This is a very thin membrane called Reisner's membrane. Reisner's membrane, very thin membrane, vestibular membrane or Reisner's membrane. And here you have a very thick, tough, fibrous membrane called the basilar membrane. Very strong membrane and very thin membrane. Okay? So these two membranes are separating the three cavities: vestibular, media, and tympana. Alright? Okay? So the fluid is not mixing. Getting the idea? Now, if the sound is coming here into the fluid system, it will travel first into vestibular. From the vestibular, it has one tawil route. It has to go and it can take the turn and it can come down and it will come here and it will vibrate the basilar membrane. Basilar membrane is the membrane most important to be vibrated actually. We'll talk about that. Okay? So the waves come in this direction. But this membrane is a very thin membrane, Reisner's membrane or the Reisner's membrane, just separating the two fluids. But it's like a very, very thin membrane. Have you seen the bag? Okay, if you break the bag and you empty it, remove the egg white and the egg yellow, okay, and look at the shell of the bag, and if you start removing inside the shell, there is a very thin membrane, okay. It's just like that. You have a very thin membrane, just separating the two fluid systems, okay. Now, when the sound waves are going from here, because the membrane is very thin, sound waves easily enter from here. They take a shortcut. They also go in a long run, but they take a shortcut and they directly strike the basilar membrane here, and the basilar membrane starts vibrating. So there are two sources of the wave traveling. Number one is through the perilymph, tawil route, and the second one is a short route, which is more important, directly passing into the endolymph, and from the endolymph we are stimulating the basilar membrane, and that's the idea. Are you getting the point so far? Yes. Is that clear? Yes. Okay. Now, I'm going for the cut section. I'm cutting the tube of the cochlea, like I'm cutting the cochlea here, and it's turning like this. So I'll see three tubes module in every cut in area, and these are the three tubes actually. The upper tube is, the middle tube is, and lower tube is perilymph, and endolymph. In this membrane, which is supporting this one, basilar, and this thin membrane is Reisner's membrane. Okay? Alright. Inside the scalamedia, over the basilar membrane, you can see there is a mount-like thing. There is a small mount-like thing. And that mount-like organ which is module is called the organ of Corti, which is module here. It is module all along the tool of the basilar membrane. It is not module in one area. Throughout the basilar membrane it is module. And if we have something like that, no, I need a page. I just need one page. Anyone, Brad? Okay. All right. Thank you very much. Okay. Okay. So I'm just giving you the idea about the basilar membrane. All right. Kind of like this. This membrane is the basilar membrane. Okay? I am making it straight, but actually it is in the coils or in the turns, if you see. An organ of Corti is module all along. If this is the basilar membrane, look again, this is happening. Okay? It's dangerous here. This is the organ of Corti module like this. And this is the remaining part of the organ of Corti, which is module. So it means all along the membrane there is a mountain range. The organ is module along the tool of the membrane. This is organ of Corti. If you cut a section from here, will you see organ of Corti?
Yes. Yes.
If you cut in the middle, will you have the same section? Here, 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 any part, you will have the same. So you will have the same appearance like this cut in this way. Look at this basilar membrane. It is made up of the fibers, thousands of fibers, and these are the frequency fibers, these sound frequency fibers. This is the starting part of the basilar membrane. Yani, I am starting the basilar membrane from here. This is the round window, and here you have the basilar membrane which is starting and going like this. Okay? The starting part, the fibers are short fibers and thick fibers. You can see the lines here. Okay? These are the fibers, like this. The fibers are going there and there throughout the tool of the membrane. Initially the fibers are short fibers and thick fibers, not easily bendable. As you go forward and forward and forward to the other end, the fibers are becoming long, taweel, very fine, slender, flexible, more elastic, and like this. Yani, for different frequencies, different fibers are modules on the basilar membrane. Imagine, okay? The whole basilar membrane is made up of the fibers which have the preset frequency. Yani, if a makhsoos frequency sound is coming here, this sound will identify the area and it will vibrate only that area where the frequency is modular, the frequency fiber is modular. The whole membrane will not vibrate, okay? The particular area will vibrate, okay? This is called the place principle or the principle of Makan. Yani, every frequency of the sound has a typical place in the basilar membrane and it will try to vibrate that area maximally, and it will try to stimulate the organ of Corti which is present over that area, that part of the organ of Corti, okay, only. Because organ of Corti is taweel, you don't want to stimulate the whole organ of Corti, you will stimulate only a little part of it which is exactly for that frequency of the sound. Are you getting this point? Okay. So this moves up and down like this when the fluid system sends the vibrations, okay? Now, look at the organ of Corti structure. I am just giving you a little histology, but that is very nice and you need that. It is important. Please look at that. Basilar membrane below. Okay? Then these are other cells. There are some very special cells, hearing cells, called the sensory receptors of the cells. Inner hair cells because they have hair, and the outer hair cells which also have the hair. How many rows of the internal hair cells do you see in this picture? One row only. One cell, behind that it is one row. One row means if this is the row of the cell, then second cell is modular here, third is modular, fourth, like this, and 1,000 cells are modular in the toll of the basal membrane, okay? For the external hair cells, 3 to 4 rows are present, 1 to 3 rows are present. Now there is a continuous row going there and going there and going. Thousands of cells are modular like that. So 3 to 4 external hair cell rows and 1 row of the internal. Okay? Internal is mainly applied for the hearing because internal hair cells are the receptors, okay, and they are stimulated by the sounds. The external hair cells are the feedback cells or the purpose of regulation or modulation of sound. They change the sound characteristics when it is transmitted, but it is for the feedback purpose. For hearing purpose, the main row is the inner hair cells. Don't forget that. Okay. All right. There are now other functions discovered. It is also concerned with hearing, but the main purpose is to support this system and to coordinate. And they also receive the efferents from the brain for protection, for coordination, other issues. Can you see the nerve fibers which are synapsing at the bases? The nerve fibers are synapsing with the bases of these cells. They are not attached, they are synapsing. Yani when the hair cell is stimulated by the sound, it will transmit the impulse into the nerve fibers. These nerve fibers are, yes, but which part? Vestibular or cochlear? It is the vestibulocochlear nerve. Which part of the vestibulocochlear nerve? You are talking about hearing. Cochlear, okay? These are the afferent fibers of the cochlear nerve, okay? And they are going into the spiral ganglion. Anyone knows about the spiral ganglion? The silence, the dead silence is telling me the story that you're not familiar with the word spiral ganglion in anatomy. Are you? No, you are not. Even if you say this, it means this. Yeah. Well, okay, let us clear because this term is coming, so it's good that we are refreshing the anatomy also. Remember the cochlea. I said cochlea, we can make it straight like a tube, and then we can coil it again like this. Okay, I showed you the turns: one turn, second turn, and third turn. Wow. When you are making the turns of the cochlea, you need a pillar or a base around which you are coiling. Okay, you need some support. If I have some tube, I want to coil it, I say, okay, I'll hold this one in the center, and then I'll make coils. Like khalas, it's easy. This is a bone, a central bony pillar. This is called modulus. What do you call it? Bony modulus. Around the modulus, you have the bony cochlea. One turn, second turn, and third turn. Base and apex of the cochlea. This is the design of the cochlea. Yani, in the markaz, what is mawjood? It is the modulus. Inside the modulus, there is a ganglion, nerve cell ganglion, where the nerve cell bodies are mawjood. It's called the spiral ganglion because it is spiral in shape. It starts from the turns of the cochlea, like the turns. So the cells are moving like this, in this fashion, they're turning. So cell bodies are collected in this fashion. So you call this ganglion as the spiral ganglion. So now you know the makan of the spiral ganglion. Where is it mawjood? In the bony pillar of the modulus. So the fibers are going into the modulus, yani where spiral ganglion is mawjood. These afferent fibers are going into spiral ganglion. Spiral ganglion has the cell bodies. From the spiral ganglion, finally you will form the cochlear nerve, and that cochlear nerve leaves. Do we have the diagram of the cochlear nerve also? Here, somewhere? Back side? I saw something. Yes, like this. So here you have the bony pillar modulus, okay, and from that modulus, spiral ganglion is there, and from the spiral ganglion, a nerve is formed, and that nerve is going out, and that nerve is your cochlear nerve, okay. This will go into the brain and will take the impulses. Did you get the design, the basic idea about arrangement? Any problem in this arrangement? Did you understand that? Now I'm just going to. Structure. So organ of Corti. Organ of Corti, on top of it there is a jelly-like membrane. Okay, a jelly-like membrane. And when the organ of Corti is moving up and down, it is brushing and rubbing against that membrane. So the hair cells are moving, or the hair cells are moving, and the hair or the stereocilia, they are bending. Okay, they have a bend. Have you done these stereocilia in the histology? You have, okay? All right. Do you have any idea that they are of different sizes, okay? Small to moderate to large, long, isn't it? It's not equal in size. This one is the largest or the longest one, and you call it kinocerium. All the cilia are attached to one another. So it means if one cilium is moving in this direction, the other ones will also move in this direction. If one is coming in this direction, all are moving in this direction. So the movement is together. It's not haphazard movement of the hair or the cilia, stereocilia I'm talking about. Okay? So the largest one is kinocerium, and they are connected by some connections. These connections are called the tip links. Tip links. At the tip, they are linked together. So you call them tip links. Tip links are connecting proteins which connect them. Okay, I'll show you the diagram here also. Look at this one. Do you see sketch diagram? This is the longest one, second one, third one, like this, and spring-like links are there, and these links are tip links. Now look at the channels. There are special channels which are mawjood here, and these channels are opened by the stretch. If one cilium is moving like this, it will stretch open the other one. The second one will stretch open the third one, so the channels are opened by stretch, by movement. Okay? Now you tell me, perilymph is like a CSF. It is sodium, potassium, other ions are mawjood. What is so special about the endolymph? Endolymph is a very special fluid, very different than a modified fluid. It is full of potassium ions. What ions? Potassium ions. Now, where is organ of Corti mawjood? Where is organ of Corti mawjood? In the perilymph or in the endolymph? Endolymph. It is dipped deep inside the endolymph. So, if these channels are open, which ion will go inside the cell? Potassium ion. So, the depolarization is caused by potassium ions, not by the sodium ions. Okay? So, potassium ions are going inside. They will cause depolarization, opening of the calcium channels, exocytosis and the release of neurotransmitter. This neurotransmitter at the base is glutamate usually. Glutamate is released and that glutamate will stimulate the afferent nerve endings and the action potential will start in the nerve ending. So it's a synaptic connection and the release of neurotransmitter is stimulating the process. So the nerve impulse is generated in the nerve fiber, it will go to the spiral ganglion, it will go to the cochlear nerve and will travel in the pathway and to the brain. Okay, did you get the idea? Okay. Now, I told you that this basilar membrane has different fibers at different places of different frequencies. In the beginning, just after the oval window when it is starting here and the round window, you have the short fibers and then the fibers are becoming taweel as you go, okay, and thin. Alright? Now you have different sounds of different frequencies. What was the range of the sound which I told you? From 20 Ashreen to Ashreen Alf, really, isn't it? 20,000, not a joke really. So, the short fibers are difficult fibers really, thick and less flexible and difficult to move. For moving these fibers, do you need a high frequency sound or a low frequency sound? What do you think? High. So it means as the high frequency sound comes, it will stimulate these fibers and this part of the membrane will vibrate. If there is a sound coming of low frequency, it will go, but it will not disturb this area. It will go to the end over there and after part of the basilar membrane where the fibers are very thin and it will try to vibrate that area. Now look at the diagram. High frequency sounds, if they are coming into the system of cochlea, are they stimulating the beginning part, middle part or the last part? Beginning part. If the intermediate frequency sounds are coming, takriban in the middle, they are maximally stimulating which part of the membrane? It's the middle part. And if the sound frequencies are really low, where will they go? They will not disturb and the last part and they will start vibrating this part. Okay? So the after part. It means there is a place principle or a place theory which says that in the basilar membrane there are thousands of fibers and each fiber is set for a particular frequency of a sound. When that particular frequency sound comes into the basilar membrane, it will locate its own area and it will try to vibrate that area maximally. And the organ of Corti which is Mawjood above that, only that part is stimulated, those hair cells are stimulated and those makhsus fibers will take the impulses to the brain and the brain makhsus neurons will be stimulated and they will identify the area or the sound as the medium frequency or the low frequency or as the high frequency sound. So there is a complete system of location in the brain. From the basilar membrane to the brain, the pathway is very specific for the sound frequency. Did you get the idea? Okay. Alright. We are done. The last part before I show you the video and then I'll discuss a few more things. Auditory pathways. Once you are in the cochlear nerve, it will go to the medulla. In the dorsal and ventral cochlear nuclei in the brain, in the medulla, on each side like this. Now, the fiber synapse, the new fibers arise, they cross the midline and they pass through trapezoid body, a structure which is called trapezoid body. Some fibers synapse in the trapezoid body and some fibers do not synapse, they simply go up. Okay? After going up, they reach another level and that level is superior olivary nucleus. Some fibers synapse, some fibers don't synapse, they further go up. When they further go up, they will pass into the area of pons and there is a nucleus called nucleus of lateral lemniscus. Some fibers will synapse, some fibers will not synapse and they will go up. The next station coming in line is a very special part called inferior colliculus and some fibers will synapse, most of the fibers synapse, some will not synapse. Fibers will further go up, next level. Next station is an area which is the most definite area. There is a guarantee that all fibers will synapse. It's thalamus. And thalamus, which part of thalamus? It's a geniculate body. And which geniculate body? It should be the medial geniculate body. The lateral is for vision, you already know that, okay? It is for the hearing. So medial geniculate body, for the first time we believe that all fibers synapse at one station, and that station is medial geniculate body of the thalamus. Yeah, okay. After that, auditory radiation, and finally they go to Mahsus part of the Markaz, that is primary auditory area, you all know, okay, the temporal lobe, that is the area, okay. Yeah.
Can you repeat the beginning?
Yes. Right from the beginning, I am again starting. Okay. Look at the pathway. I'm not going into the details of the pathway, but I am telling you from the cochlear nerve which is formed. The cochlear nerve fibers enter into the medulla, lower medulla, where dorsal and ventral cochlear nuclei are modular. Most of the fibers synapse, but not all fibers synapse. Okay. Most of the fibers synapse. Or some book says that here it is the station where all fibers synapse, and it is the station where all fibers synapse do place. But usually this is the station we are shown about where all fibers synapse. Anyway, fibers synapse, most of the fibers cross over to the opposite side, passing through a structure called trapezoid body, a gray matter structure. Some fibers synapse in trapezoid body, some fibers don't synapse, they pass to the opposite side, they ascend. Now they go to the upper level of the medulla and they will go into superior olivary nucleus. Okay, they will synapse, some will not synapse, will further go up into the pons area where another small nucleus module, nucleus of the lateral lemniscus, some synapse, some don't synapse. They go to the next station, inferior colliculus, in the midbrain, some synapse, some don't synapse. Then they go to a station where all will synapse. That is your medial geniculate body. After that synapse, the fibers finally run up in the form of radiation and they go to specific area, primary auditory cortex. It is the superior temporal gyrus, and then from there it goes into the association areas of secondary and tertiary for the understanding of the area. Is that alright? That is a simple approach about the pathway. Are we comfortable with that? Now please, before I do anything else, I would like you to, and everybody will be silent, pin drop silence, because we don't have the speakers available. It's all about hearing, please. So you have to listen. Be silent.
The sense of hearing is accomplished by a process known as auditory transduction. The ear converts sound waves in the air into electrical impulses, which can be interpreted by the brain. As sound enters the ear, it passes through the external auditory canal, where it meets the tympanic membrane. The tympanic membrane then vibrates in response to the sound. Sounds of a lower pitch or frequency produce a slower rate of vibration. And sounds of low volume or amplitude produce a less dramatic vibration. Higher frequency sounds produce faster vibrations. The tympanic membrane is cone-shaped and articulates with a chain of 3 bones called the auditory ossicles. They consist of the malleus, the incus, and the stapes. The movements of the tympanic membrane vibrate the ossicles, passing on the information of frequency and amplitude. The 3 bones pivot together on an axis shown here in red. The pivotal axis is due to a series of ligaments which hold the bones in place within the middle ear cavity. The anterior malleal ligament and the posterior incutal ligament are of particular importance for the pivotal axis. 2 structures which normally obscure this view of the middle ear have been removed. They are the chordae tympani nerve and the tendon of the tensor tympani muscle. Through the ossicles, the vibrations of the tympanic membrane are transferred to the footplate of the stapes. is filled with a fluid called perilymph.
The movement of the stapes, you saw the piston-like movements, back and forth movement going inside. This is one